Welcome back. Our next company is IRLAB, a Swedish biotech focused on the development of new therapies for patients living with neurological diseases. With us, we have here Nicholas Waters, the CEO. Please, Nicholas, the word is yours.
Thank you very much, and nice to be with you again. And thank you once again for this opportunity to present IRLAB. Company focusing on Parkinson's, novel treatments for Parkinson's disease. Just take you through the disclaimer and just a brief overview of the some recent data we presented in the Q1 report, indicating that IRLAB has a very strong cash position, which is a prerequisite in these times, which is also in alignment with our long-term strategy. At present, we are investing in two phase 2B programs. So next step for both of those assets will be phase 3. And we have also broadened our pipeline during the past year, and we are now investing in preclinical development of a number of assets. I'll come back to that towards clinical phase 1. As you can see, we have on the left-hand side of the slide, we have thoughtfully built up a cash position over the past 3 years. And since we entered into the Nasdaq Stockholm, we have continuously increased our number of employees from around 12 to 15 to now 30 people. And of course, the cost structure grows with the number of employees, but most of all with the development of the pipeline. We have prepared for this and are now moving forward with our assets in Parkinson's disease. Just a few words on the symptomatology of Parkinson's disease, which is a complex neurological disorder depending on loss of dopamine neurons in the midbrain, which affects basically all aspects of life for patients living with Parkinson's disease. First, typical signs are the bradykinesia, the rigidity, and tremor, which are the hallmark symptoms of Parkinson's disease. In that aspect, we are now developing a totally novel strategy for supporting treatment of these symptoms in PD. We are focusing on once daily treatment, which differentiates from today's treatments, which are up to 9 to sometimes even 12 times a day. Patients need to take their levodopa treatments. So we're looking forward to a quite significant shift in the simplicity of treating the early stage symptoms with this strategy. Next symptom complication that comes into the algorithm here is the dyskinesia, the motor fluctuations, and the psychosis symptoms that these patients or individuals experience. There we are developing right now mesdopetam, which is in phase 2B. We are moving this asset forward together with our partner Ipsen. And this is a strategy to optimally treat dyskinesia, but also psychosis. Later on in the development of the disorder, there is a loss of cognitive function, cognitive impairment, which is usually coupled to dementia, and also apathy, confusion, and depression. Common symptoms in mid to late stage Parkinson's disease. There we have our preclinical assets 942 and 757 addressing these symptomatologies via modern new, or as I say, totally new mechanisms of action. And at the top, one of the biggest problems in Parkinson's disease, which is balance and falls. The loss of balance, which leads to falls, which leads to fall injuries, which is very costly for the patient, but also for society. And here we are developing pirepemat, which is now in phase 2B for this indication. So we are filling up the needs here in PD with a quite comprehensive pipeline. We are using our advanced systems biology strategy, which we have named ISP, the integrative screening process. This is a system where we collect we use phenotypic screening strategies, collect a lot of data, analyze them by means of machine learning, artificial intelligence strategies, to define structures and programs. We can also predict usefulness of any new drug. And this highly efficient system, it results in the discovery of first-in-class compounds, which is very important from a competitive standpoint. We also get very strong IPR. We discover molecules which would otherwise go undiscovered by traditional strategies. And strong IPR is once again very important for the competitive edge that we have with our programs. As I said, we can predict which indications are most suited for any new chemical. And we also see, going back in time, around 20 years where we've used ISP, that we see an improvement in the likelihood of phase transitions. And this is one of the biggest issues in drug development today, and it's been discussed a lot in the literature. How to manage the attrition, or how to reduce attrition in any pipeline. And with ISP, we discover and develop molecules which have a higher likelihood of surviving phase transitions from preclinical to clinical stages, and within the clinical development pipeline, from phase 1 to phase 2 and to phase 3. Looking at the numbers we have generated based on our own CDs over the years, we estimate roughly somewhere between 2 to 3 times higher likelihood of reaching phase 3 with an ISP-derived molecule than with the standard industry documented numbers. This, we think, is also adding to the competitive edge that we have. Slide 7 illustrates the pipeline as it looks today. We have broadened it over the past year, as I said, going from the bottom with the P003 program, 757 and 942, preclinical assets now in preclinical development towards IND. And then we have pirepemat for balance and falls, and mesdopetam for dyskinesias and psychosis. We have a differentiated strategy, commercial strategy for these assets, with showing the partnering with shown by the partnering with Ipsen last summer for mesdopetam. This was a $363 million deal with double-digit royalties on top, which we are very proud of course. And we have a very, very strong partner in this program, dedicated to developing mesdopetam to the market. We envisage a similar strategy for pirepemat, that is, during phase 2 or after phase 2b data, we expect to join forces with a partner. For the early stage molecules, the preclinical assets, we are discussing the possibility of actually reaching early stage partnering possibilities here. So we have a quite varied strategy towards partnering with our pipeline. Looking at the news flow over the past coming years from each of these assets in the pipeline, and this is just illustrating, slide 8 here, just illustrating the news flow from the pipeline, that is excluding any other major events such as deals or other types of events. So in the mesdopetam program, we expect to see data late this year. We expect to see phase 3 initiation next year, and also a broadening of the indication panorama for mesdopetam. The marketing preparations will start also in 2024, but that's something that basically starts already when you're working in the preclinical development. And then for pirepemat, we have just recently initiated the phase 2b study. We expect to see data by the end of 2024. By the end of 23, we'll see top-line data for the ongoing phase 2b study across Europe. And similarly, we expect for the early stage compounds to reach clinical trials within 23 and 24. So this is a quite rich news flow for a smaller biotech company like ours. A few words on pirepemat, and the unmet needs out there are huge. About 45% of all people with Parkinson's disease, they fall recurrently, up to several times a week. And each fall imposes a risk of severe fractures and hospitalization. Pirepemat is a molecule which we have designed to improve those systems in the brain which govern balance and have the potential to reduce falls. Just looking at the societal costs for these falls and the fall injuries, around $30,000 per patient for a patient ending up in hospital. This is an untapped market. There are no current drugs registered for reducing falls in PD, and it's a huge market. Many patients across the globe. The study is designed as a 3-arm study, 1 placebo arm, 2 different doses, 300 and 600 milligrams. We are looking at a 3-month treatment period after 4 to 6 weeks of screening. That's following the patients to make sure that they fulfill inclusion criteria of at least 2 falls during the past or the 4 weeks prior to randomization. And the primary endpoint in this study is, of course, to evaluate effects on falls frequency. We are proud to have been able to start this study despite the situation over the past year around Europe with the COVID and now also the Ukrainian situation. Looking at the regulatory status here, we have received approvals from all the countries we have applied to start the study in now. France, Germany, Poland, Spain, and Sweden. So this will initially be a European effort. And we will focus now on working with the hospitals and the sites and doctors who will be involved in the study, including the patients, of course. Looking at the preclinical assets, we have 3 or at least 2 programs, now 2 CDs from the first program, P001. 942 is a molecule specifically designed to improve cognitive function and brain health in neurological disorder. This means that we are looking at potential to broaden our scope a little bit outside of Parkinson's disease as well. But of course, Parkinson is our main target. The problem here with cognitive decline in neurodegenerative disorders is that there is a disruption of frontal cortical neurocircuitries, which we can resurrect with the treatment with 942. And here we see potential for both symptomatic relief, but also for disease modification. The status right now is that we are right now developing the CMC. We are moving towards regulatory studies necessary for filing an IND during next year. With 757, we are moving towards an indication where there are no treatments today. That's apathy. This is a huge problem in neurological disorders. Around 50% of all patients with neurological disorder are at risk of developing apathy. And it's a huge problem for them and for their caregivers. The pathophysiological background or the mechanism underlying this is quite similar to the ones related to cognition, but there is a disruption of frontal subcortical circuitries, and those can be treated or improved based on preclinical data that we have generated, be improved by 757, which increases the neural activity in these regions, specific regions, and in the communication between the regions. Also here we have potential for symptomatic relief and disease modification. Similar to 942, we are now running a CMC development campaign to be able to apply for, and toxin safety studies, of course, to be able to apply for phase 1 during next year. P003, it's the latest addition to our pipeline. We have worked on this for a couple of years. We have now we're looking at a totally new strategy to treat the basic symptoms of Parkinson's disease. And one of the problems with Parkinson's disease treatments available today is that they impose a very strong fluctuation in symptomatology. There are overshoots during certain periods of the day, and there are troughs where there are no effects of the treatments. This, we think, can be resurrected by a long-lasting treatment like the ones we are developing in P003. So we think we have potential to provide an optimal functional efficacy without limitations that you see with current treatments. The mechanism here is similar to what is used today in the gold standard treatment levodopa. That's a full dopamine D1, D2 agonist strategy, but with a much improved pharmacokinetic property. We are looking at two different strategies. The first generation, we are now in lead optimization. We have previously said that we expect to see a CD from this program this year, which will bring yet another candidate drug into development in our broadening pipeline. And then we have a second generation strategy ongoing on the drawing board right now. A few final words on the collaborative project with Ipsen. That's the mesdopetam program. This is a program which is now fully financed through phase 3 to market globally together with Ipsen. We are looking at improvement of motor function through prevention of reduction of dyskinesias. We have generated data which support the potential prevention here as well, besides the just symptomatic treatment. But there is also a possibility of treating psychosis in Parkinson's disease. There are reasons to believe that this mechanism has antipsychotic properties in Parkinson's disease as well. 30% of all Parkinson's patients have problems with dyskinesia. An additional 30%, 35% have problems with psychosis. Right now, we are running a phase 2b/3 study across Europe and the US. We are recruiting in both these geographical regions. Preparations are ongoing for phase 3 studies, and we are supporting Ipsen's efforts to bring this asset to patients. Besides LIDs and psychosis in Parkinson's disease, there is also an opportunity in tardive dyskinesia, which is actually an even bigger indication globally. It's a side effect of antipsychotics, which can be treated with D3 antagonists, which is exactly the mechanism underlying the efficacy of mesdopetam. So, since we introduced IRLAB in 2017 on Nasdaq, we have now full validation of our strategy towards our vision of being a sustainable deliverer of new treatments for complications in neurological disorders. The science has been published in highly ranked publications over the years. We have had quite good successes with our translation work from preclinical to phase 1 to phase 2 and to phase 2b. The innovation and the novelty is proven by the unique INN names that we have been provided, mesdopetam and pirepemat, indicating they represent totally new classes of CNS disorders, treatments for CNS disorders. And then the commercial value of our work has been validated by the global licensing of mesdopetam to Ipsen last year. So, with those words, I would like to just conclude that we are on a path to deliver strong growth. We have the resources in-house right now. We are building on our competence base, and we will continue to do that during the year. Add commercial competence during the fall. That's our plan. Thank you very much.
Thank you very much, Nicholas, for your nice presentation. I just wanted to remind you if you could turn on your camera.
My camera is on, but I am also showing the... I'll take away the...
Great.
Oh.
Thank you so much.
Can you see me now?
Yes.
Good.
Thank you so much. Now we have time for a few questions. And I will start talking about your interesting approach for drug discovery. It's based in phenotypic screening. And for someone that doesn't understand what phenotypic screening means, could you give a brief explanation on what does this represent and how this compares with traditional screening?
In traditional screening, you start by defining the target you want to address. And then you look for molecules which have properties that can modulate this target. And that depends on a very long period of defining the target you want to address. In our case, with phenotypic screening, which is growing in the industry, is that we allow the living system to inform us about the global effect of any treatment strategy. And the ISP system supports us in discovering drugs which have beneficial effect at the system level, not only at the target level, but also at the system level. There is usually a disconnect between a certain target and a system level effect, and we are capturing the system level effect as a first thing in our discovery. And that means that we don't need to synthesize so many drugs to come to a conclusion of a CD, and we have a very rapid discovery process. It's about half the time in preclinical work compared to a traditional strategy. And that halving of that time is actually very important for the value of the program and the time you have as an exclusive on the market.
That's very, very interesting. And my next question is regarding your newly announced molecule, 757.
Yes.
And you're targeting a space where right now there is no solution. These patients have no solution for apathy. And
Exactly.
How come that there is no solution right now? Is it because the pathways that regulate this symptom are too complicated to target?
I don't think so. I mean, if one looks back in history, a number of currently available drugs have been tested with quite bleak results. But using ISP, we have defined and the literature, of course, the scientific literature behind the pathophysiology. We've used ISPs to actually define what the molecule should do. And when we do that, we have the tools in our hands to actually discover molecules which addresses exactly the right mechanisms. So I don't think it's impossible. It's that difficult in a sense, but of course, you need the tools that we have developed to do that.
Yeah, I understand. Thank you. And one last question regarding mesdopetam. As you previously mentioned, together with other key opinion leaders, mesdopetam has the potential to also have an impact not just on the treatment but the prevention of PD leads. Could you give us a little bit your view on that?
Absolutely. And the view is based on preclinical data which we published at Neuroscience a year ago, where we have looked at chronic treatment with mesdopetam in animal models of dyskinesia. And what we can see is that we do see a prevention of the effects of levodopa. Levodopa is what drives the dyskinesia, basically, in patients and in those preclinical models. And it seems that we can actually block that effect over time. So long-term treatment with mesdopetam, hitting the mechanism responsible for generating dyskinesia. If we do that early, we can probably prevent the development of dyskinesia with mesdopetam.
That sounds very, very interesting. Nicolas, thank you very much for being here with us today and for your nice presentation.
Thank you very much for having us, and always a pleasure.